Okay, proceeding to part 4. So at this point, let me go here. So what have we decided? Let me go back to what have we decided? We decided that the female 0 is either in cis double mutant or is in trans double mutant, and female 1 we decided was in the trans configuration. Female 2 is only heterozygous at the orange locus, and female 3 is in the cis configuration. And the male in question, so there are multiple males with multiple males, and the male used here that was black and was either this or, if X-linked, this. And I don't think I cannot figure out how we can distinguish between, I don't think there's enough information to distinguish between those two possibilities. What did I do there? That's something else. Okay, so now the question, so explain why none of the crosses show identical phenotype frequencies. Okay, because we just showed them all that they're different cis trans, and that. State whether each mutant finite dominant or negative recessive, we did that. Or loci for light orange linked, they're linked, and if they're linked, state whether X-linked or autosomal. I don't think we can distinguish it. And draw a map showing the distance between them. Okay, the distance between them would be, well, what's that? How many are recombinant here? So we can add up all the parentals, that's around 80, and what's that? That's less than 10. So we have about, I don't know, it looks like around 10%. This doesn't tell us, only the recombinant ones do. And here again we have around 80 and we have around 15. So something less than 10 map units. I don't care to do it precisely. Diagram the linkage arrangement of the loci for the light orange eyes and black bodies in each of the 4 females. So we did that, except we can't distinguish also mother 0. Okay. Now this was the one I wanted to do it on, the 1415, to identify the location of the light orange eye color described in question 1414 quickly, and STR, short tandem repeat analysis were performed. DNA was isolated from light orange eye female 2, the red-eyed black body male to which is mated, in each of their 140 gray offspring, and a panel of STR markers analyzed in each DNA sample. They do something funny here. Okay, so I don't know if I'm going to go through all of this. One is that now that the genome is sequenced, and sequenced not just for melanogaster but also for pseudo-obscura, we don't do this. This kind of mapping, of which the book has a lot of, it's funny for me in that I want to make sure students understand meiosis and how things work, and how little we know, what part of it we understand, and also what part we don't understand. And this technology of short tandem repeats and markers and mapping, this was hardcore molecular genetics before the genome was sequenced, which happened, I think, after 2000, maybe around there, at the earliest late 90s, and pseudo-obscurans is also sequenced probably later. The thinking is useful, but we don't do it that way. So the orange-eyed female 2 and the black, and then the one thing that they do here is, let me continue this so I can show you why it's a little strange. Each was analyzed in a DNA sample. The results obtained with 4 representative STR markers are shown in table 4. 14a. Which marker shows evidence of linkage to the locus for orange eyes, which do not, and for which is impossible to evaluate? For any STA marker shows evidence linkage, what is the map distance between it and the locus for light orange eyes? What is the linkage arrangement in female 2? So female 2 here, so what we're looking at is female 2, and we're female 2, that's the, did I get this wrong? I'm going to pause for a second here. Unpausing. Female 2 is the boring one. Yes, it says so. Clearly it's not me. Female 2 is the boring one. So, so there's something funny about this problem. So we wouldn't, so let's assume, as the book does, that it's autosomal. So we're looking at this cross, and we have 140 offspring. And we have a bunch of, I didn't show you yet, we have these SDRs. Okay, so what's a short tandem repeat? Maybe it's something like AG, tandem, right? Two of them AG, AG, like this, and we count how many repeats are there. 1, 2, 3, 4, 5. So the information is, we determined that the genotype to the parent, the female, was at this. Okay, so what's an STR? So there's some, imagine some DNA, and there's a locus, perhaps there's a locus of interest here. I'm going to use different than O and B. Let's say it's C and D, where there may be some STR here, there may be some STR here, there may be some STR here, there may be STR on a different chromosome. And as a result, if we can see whether this STR co-maps with this, and just like any other allele, we just measure instead of looking at the fly and seeing that it's black or gray or orange or red. We do a PCR because we know the genome in this area, and knowing the genome here and here, we can make little primers here and see how long they are and run them, so we can isolate the DNA of each of these offspring, or any number of offspring, and we can do PCR and run them on a gel, or capillary electrophoresis would be modern method, and see how long they are. So we're going to have different STR numbering, so that could be 10, 11, 12, 13, an identity, and how long they are is going to be different. And because each organism is diploid, that's the other thing why here we finally have evidence. We finally have evidence that these that they're autosomal, because the male in some of these cases. Okay, there's something interesting here. I'm going to. The male in some cases has 2 different markers, even when it's linked. Okay, we haven't talked about that yet. Okay, so they're diploid, so they're going to have, for each STR locus, they're going to have possibly 2. They could be the same, and because these are nearby, possibly nearby an allele interest, we would expect that the divergence, this gets a little bit complicated because we have to think about the divergence of the allele. So let's say for orange, once upon a time everything was wild type, there's a mutant orange, where also at the same time there was an STR, and that STR may have diverged to be different here. It could have diverged beforehand, it could be somewhere else, it could diverge afterwards, in which case they're going to be different, and the recombination between the STR marker and the locus of interest is not at equilibrium, and it's going to be associated with it. Okay, this is back for earlier chapters. I don't want to spend a lot of time on this one because I think it's not very interesting, but let's look at this. So what can we see? What markers show evidence of linkage and which do not, and of orange eyes? And notice that we chose the female, for which there's only orange. So in a sense, the male here, we don't care about the black body anymore. We're only looking for the O. So where the male is a test cross here. We're seeing a heterozygous female crossed to a homozygous male, and the data here don't talk anything about black eyes. Okay, so here what we see is that the STR marker, 8 and 8, so she has the same number of repeats from both parents, so that's a clue right away. Maybe it's not linked, it's on a different chromosome. Would kind of expect to be different because we expect it where she is heterozygous at O, so we'll kind of expect to have different STRs. If it's linked, it's not. And we see that it doesn't matter. Orange-eyed, red eyes, there's no segregation. Here, STR2, aha, we see that orange comes with the orange comes with so what are we looking at here? So genotypes of progeny, that's 6 comma, that's a 6 comma 5. So 6 from the mother, 5 from the father, 4 from the mother. So we see that 6 associates with orange and 4 associates with red. Here we see no segregation. Here we see each of the parents have 2 different markers, but we see nonetheless that we see an association of orange with 3 and red with 6. So maybe it's the same 6. We would expect it to be the same 6. That's more likely. So 6 with orange. So we can say 6 STR. That's unrelated. STR2. 6 correlates with 6 correlates with orange. So 6 here, orange, and STR2 with 4 relates to red, and at STR4 we have also correlation of, orange is with 3, and those are unrelated. They're different. STR orange and STR4 with number of 6 is 6 repeats is red. Okay, so can we map them? No, because they did something. They're showing us an experiment that's kind of funny. If someone's going to do the flies, they're going to do all of them together, and we don't have now with the number of progeny. They did this individual. They say there's these are the kind of progeny, and they have this, but they don't tell us the frequency. Which of these 33? Let me do it here. Which of these 64 and which of these 6 are also these 4 and these 9? They're not telling us, and they would have had that information. So it's a little bit funny. Okay, so what can we do here? I'm going to speed up a little bit. So STR's loci 2 and 4 show linkage. The other 2 do not. We can't. So there's nothing we can say. For that shows that what is the map distance? We can't map because they didn't collect the data. All we can do is we can map. We can map. We can't tell the. We can only say that something. We can say the genotypes here of the orange with STR4 and 3, and the orange with STR2 and 6, but we can't map 2 and 4 next relative to each other. We can only say, the map distance is going to be, okay, it's about 10%, 10 map units between STR, a little bit less, or the map distance here is more like 20%. So, and the book has some numbers that they added wrong or something. Okay, so all we can say then, and I'm going to run out of time to do the proper notation. All we can say is that STR2 and STR4 are linked to the orange, and we can see the map distance between STR2 6 repeats and STR4 repeats to orange, and STR2 with 4 repeats to red, and STR4 with 6 repeats to red, but we can't see the relative distance. We can't map them which order they are relative to each other. Okay, I'm going to stop here. I don't care that much about this particular problem. This problem is rather difficult because one has to constantly consider possibilities. That's what makes it nasty. Oh, is it recessive? Is it dominant? Is it linked? Is it not linked? Is it X-linked? That's what makes it difficult.